So, chapter 16, we get to talk about amino acids, proteins, and enzymes. So, many physician's assistants take on the role of primary caregiver. Their duties include obtaining medical records and histories, diagnosing illnesses, educating and counseling patients, as well as referring patient when needed to a specialist. So, they are helping out the physician a whole bunch. So, proteins and amino acids. Protein molecules, when compared with many of the life compounds we have studied, can be gigantic. So remember, a lot of the things we've been studying are micro. Well, amino acids can be really big, okay? So for instance, one thing that you can actually physically look and see and feel is the horn of animals are made of proteins. And so that's a really big one on this longhorn cow. Okay, so we are going to learn how to classify proteins by their functions, give the name and abbreviation for an amino acid, and draw its ionized structure. So proteins in the body are polymers that are made from 20 different amino acids. They differ in characteristics and functions, and that depends on the order of the amino acids that make up the protein. They form structural components such as cartilage, muscles, hair, right, and nails. They function as enzymes to regulate biological regulations, as in digestion and cellular metabolism, as well as hemoglobin and myoglobin. They're functional, right? They're transporting and storing oxygen in the blood. So I always say our DNA is what codes for things, and then the protein are what's functional in the body. Okay, so we can have the DNA for it, but until it's expressed into a protein, it really doesn't matter, right? Because the proteins are the active guys in the body. So here are some different structural classifications of proteins. So you have structural proteins, contractile proteins, transport, storage, hormone proteins, enzymes, and protection. So this is a great little summary. It says they're structural. Think of it as like scaffolding, right? These are your collagens and your keratins. You have transports. They're going to help shuttle things and carry things through the body. And that would be like hemoglobin transporting oxygen, and lipoproteins transporting lipids and fats, right? Your hormones like insulin, glucagon, growth hormone, all those things. So these are all just very important structural classifications of proteins. So amino acids are the molecular building blocks of proteins. So they have a central carbon that's called the alpha carbon, and this is bonded to two functional groups. One is called the ammonium group, and the other is called a carboxyl group or carboxylic group. So they also have that central carbon bonded to a hydrogen atom and an R or a side chain, in addition to the carboxylic acid or the amino acid group. So I call this the carboxyl group and the amino group. Okay, so the way that I look at this is it's kind of like an ant body anatomy, right? And I know that's really silly, but you have the head, you have the body, and then you have the butt of the ant, and then it has two arms. One is going to be just a hydrogen, and then this R group, R is going to be variable. So remember when you took stats, right? R squared is variable squared. And so R is the variable. And what does that mean? That's going to be what's different between each of the different amino acids. So the backbone is going to be the same with slight modifications. And we'll talk about that depending on the pH. We can drop or add hydrogens here, right? But the R group here is what's going to change. And otherwise, the backbone is going to be pretty consistent. Amino acids can be classified either as nonpolar or polar. So the nonpolar is not polar. And another way to say it would be that it's hydrophobic or scared of water. And those are the ones that have hydrocarbon side chains. So break those down into your words. Hydro is H and carbons are your carbon, right? So your hydrophobic amino acids are ones that are just going to have carbons and hydrogens on the side chain. And basically what we really care about is who's on the very outside of it. Then you have polar or hydrophilic, water loving. And these are polar with ionic side chains, okay? So you have that backbone again, see how the backbone's the same, but then on the outside, the very outside of the R group, we have an N, and that would make it polar. So for instance, valine nonpolar because it has carbons, asparagine is polar because it has that nitrogen. So here is a group picture, everyone get together and smile, of an amino acid. When it's nonpolar with the R group, it has an H, it's alkyl or aromatic. Okay, so here is the picture of, glycine, alkaline, valine, right? So you don't have to memorize all these, but what I do want you to be able to do is look at a picture, say, okay, that's the same, that's the backbone. Let's look at who's on the very outside of the R group and see, oh, okay, these guys are going to be carbons, so those are going to be hydrophobic nonpolar. Just to point out here, and we've already talked about this, but when it's just a ring like this with no letters in it, then that's going to be just a ring of carbon. Okay. Then we have our polar amino acids, and this is when the amino acid is polar, when our group is an alcohol or a thiol or an amine. Okay, so you have oxygen, right, sulfur, or nitrogen on the outside, and those are going to be your polar amino acids. So the structural formula in writing it, the amino acid has that alpha carbon there in the center, and it has the ant head and the ant butt, right? So that means the amino group, the carboxyl group, and then that side hydrogen group all attached to that alpha carbon. And then the fourth component is the R group that changes and differs for each of the amino acids. And it either has a 3-letter or a 1-letter abbreviation that is made from its name. Okay? So how do we draw these guys? Well, all amino acids are going to have that amino and carboxyl and hydrogen on the alpha carbon. And again, the only thing that's going to differ is going to be that R group, all right? So when an amino acid is acidic, it's going to have the R group has the carboxylic acid. Okay, so you can see here, the difference here is going to be a length in the side arm, and the R group is the same, and so the R group is the carboxylic acid as well. So it's going to look very similar to the backbone because the carboxylic acid carboxyl group, right? It's COOH versus COO minus. So this one, the R group, is the same as the backbone, but don't get them confused. Okay, but it's going to be acidic when that R group is the carboxyl group, or otherwise known as the carboxylic acid. And so since acid is a root of the word, we know that it's acidic. Then we have basic amino acids, and this is when the amino acid is basic when that R group is an amine group. Okay, so look, we have the nitrogens and nitrogens, right? So when it's the amine group with the NH2 or the NH3+, that's going to be where you have your basic amino acids. All right, so let's practice. So let's identify each of the following amino acids as polar or nonpolar. So pause me and come back. Okay, so this glycine is going to be nonpolar, okay, where this threonine is going to be polar. So what are we going to do? Let's look and see where the backbone is. Okay, and glycine's a little tricky because his sidearm is only hydrogen, right? Where this guy is polar because of the oxygen. All right, great. What about this guy? Draw the ionized form for each of the following amino acids and write the 3-letter and 1-letter abbreviations. So pause me and come back. All right, so is this what you got? So valine is going to be your backbone and your sidearm, and the abbreviation is either VAL or V. And cysteine is again your backbone with the sulfur hydrogen. Cysteine says kind of special amino acid because he's the only amino acid that has sulfur on the outside. Okay, so he actually forms a special bond called a disulfide bond, but we'll talk about that later. And the abbreviation is CYS and C. Okay. So another way that you can look at these too are those stereoisomers. So it's basically the same thing except for the location, right? So all the alpha amino acids except for glycine are chiral. So remember that was the mirror image guy. The alpha carbon is attached to 4 different atoms, and the NH3 group is going to appear either on the right or the left of the chiral carbon to give it either the D or the L enantiomers. Okay, so just remember this one's on the left, this one's on the right. So the way I remember the L versus the D is left L, right? And then D would be the right. So just remember wherever you place it, it will affect the naming, the naming of it. All right, so let's see with this one. Pause me and come back and figure out which one is D or L. All right, this one is an L enantiomer. where this one is D-serine. All right, good job, you guys.